So you can prevent 80% of the cases by simply administering folic acid. And it's not just during pregnancy. If there are any childbearing people in the audience, it may well be very important to have it at conception, and we can talk about it privately if you wish. The same story may turn out to be true for MS. So this is Glasgow. This is a totally separate, independent population, and here what you see is this striking distortion of month of birth. So this is compared to the general population, and April is the peak. Interestingly enough, it's an April peak in Glasgow, whereas it's a May peak in Edinburgh. And then the November dip is the same. And we'll go on here now to Dundee. And in Dundee, it's a June peak. So you're seeing that within the microgeography of Scotland, the peak month of birth effect, which is very large in all three, varies by a month or two, which is probably telling us something about the local conditions. And their dip is actually September. So we see variation both in the peak and in the trough, suggesting they may actually be not simply opposite sides of the same coin, they may actually be slightly different. So we reasoned that if the month of birth effect was familial, which it was, and it was part of the gene-environment interaction, it might be within the MHC, which is the dominant risk region for MS, and in fact, it is. So the people that have this gene are the ones who are responsible for this month of birth effect. And this very loudly says there has to be gene-environment interaction at this locus, and as we'll show you in a moment, it turns out to be epigenetic. And I'll tell you what that is in a minute. Now, some of you might have seen this slide before because I've shown it on and off over the years, but this is an extraordinary family, 2 hours north of Winnipeg on the Peguis Indian Reserve, and the mother and father are in the center. The father's on the right there, and his wife is on his right, and he is the son of the Indian chief, the Peguis Reserve, and she was the daughter of the missionary sent to convert them to Christianity. Now, they had 18 children, and you can see some of them look like mom, some look like dad, some look like in between. And the question here is, what about MS? Well, her family came from the Orkneys, and they were part of the Hudson's Bay Company. I don't know if any of you know the history of Canada, but the Hudson's Bay Company was heavily populated by Orcadians. And he comes from a population where MS virtually never occurs. So the question here is, what happens in their offspring? You have a susceptible strain crossed with a resistant strain, if you like. We were talking about a mouse experiment. And in fact, 6 of the children got MS. And it turns out it matters who the susceptible parent is. We've done other studies now in various populations, and it's more important that the mother be the susceptible ethnic background than it is the father. This is the first hint that that was going to be the case. Now this is the next generation in that family. So in this generation, we had 6 of 18 who had MS. There were 150 people in the next generation, and MS basically all but disappears. There was one person who might have had MS, but otherwise no MS. Now, what kind of inheritance pattern is that? Well, I'm going to tell you in a moment. So, the evidence gradually has accumulated that MS is very strongly influenced by epigenetic effects. We told you the mother contributes more risk than the father, which tells you that there has to be something special about the mother. If you look at cases where an aunt is affected and a niece or a nephew is affected, you can ask, which is the connecting parent? That is to say, if the aunt or uncle is affected, is it the father of the niece or nephew, or is it the mother of the niece or nephew that's connected to the affected aunts or uncles? And we find the same thing, which is that, once again, it's the mother that's the one that's affected. So that's an interesting story. Within a nuclear family, the inheritance of MS looks like an autosomal dominant pattern. So the risk to your child, to your parent, to your brother or sister is all about the same. It's something like 3 or 4% in northern populations. As soon as you move away from the nuclear family, it changes drastically, like I just illustrated. So that's a really interesting and relatively novel inheritance pattern. Now, there are a number of other things listed here which are slightly arcane. I'm going to skip them, but I'm going to tell you how we went on to the next step to show that it was really going to be epigenetic. So we reasoned that if the rate of MS has changed, and by the way, the rate of MS has changed spectacularly in some places. In Scotland, the sex ratio was 1-to-1 in 1950, and now the sex ratio is almost 4-to-1. So it's tripled or quadrupled in 50 years. That has to be environment, at the risk of stating the obvious. Anyway, we knew the rate of MS had changed in Canada. I'll show you a slide of that. And we knew it was gender specific. It was almost entirely occurring in females. And we were suspicious that the risk was determined by a gene-environment interaction or something epigenetic. And so what we did was, I'm afraid it's a little complicated, but we looked at the allele frequency of the genes, the main determinant of MS risk, or haplotype DRB1-1501, and we asked whether or not the frequency in nieces was the same as in their own aunts. I hope that makes sense to everybody. Anyway, if there's been a modification of this gene region by the environment, which we were strongly suspicious of, then in fact the frequency of the allele, the frequency of the 1501, would be greater in the nieces than in their own aunts, and that's exactly what we found. So this is defining epigenetics for you. In the 40s, it was defined as a branch of biology studying the causal interaction between genes and their product, which bring the phenotype, which is what you see, the manifestations of the individual, into being. And more recently, a more modern definition has changed the gene function. They're meiotically heritable, and do not entail a change in DNA sequence. So a lot of people had the idea that we would sequence everybody's genome, and that'd be it. We could close our shops, and all knowledge would be emanating from that. Well, that turns out to be incredibly naive. And sequencing genomes has actually provided amazingly little information for most of the common diseases of mankind. Amazingly little information. Well, we were thinking about vitamin D. We knew that the MHC was the main gene region. So then the question was, how can we link the two together? And one obvious way was to see whether or not this might be regulated by vitamin D, because vitamin D turns out to be a transcriptional factor which regulates a lot of genes. Unfortunately, it's called a vitamin, which is bad, actually, because vitamins, as soon as you mention the word vitamin, you think of kooky people and Linus Pauling and preventing colds with vitamin C and giving people cancer with vitamin A and vitamin E. And it has a bad connotation, but in fact it shouldn't be called a vitamin at all. It's really more a hormone. You make it yourself, which immediately contradicts the basic requirement for a vitamin. And it's primarily derived from sun exposure in your own. And we've evolved to deal with it for millions of years. We've evolved to deal with it. So worrying about taking a little too much vitamin D is absolutely ridiculous. For a million years, we're running around in the plains of Africa naked, and in fact, our bodies had lots of time to learn how to deal with vitamin D. So it makes more sense to ban holidays to Portugal than it does to prevent people from taking vitamin D in the recommended amounts. Anyway, we had a big surprise. Here we were looking at the promoter region, which is the area that tells you what regulates the gene. And here it is, the main gene region that makes you susceptible to MS, and immediately adjacent to the gene is in the classical promoter region is a place to which vitamin D binds. It's called vitamin D response element, and we showed that it was functionally active. And a lot of sequencing was done by one of our very hardworking postdocs, and it was very conserved. That is to say, 1,200 chromosomes in MS patients were sequenced, and this little vitamin D binding site was present in every single one of them. And this has been present probably for 40 million years. So we now had three pieces of information that says the environment is interacting with the genetics. The first was that the main gene region was regulated by vitamin D. The second was the evidence for epigenetics, that is to say that something is clearly happening in passage through the mother of this gene region. And the third part was that the month of birth effect was localizable, once again, to the same gene region. So here are three unambiguous bits of evidence that the main gene region that's disposing of MS is acted on by the environment, sometimes temporarily and sometimes permanently, to alter the function of that gene region. Now, if we had this by itself, this would be almost prima facie evidence that vitamin D would be implicated. Because vitamin D is a cofactor for epigenetic modification, and the seasonal effect of month of birth clearly links as a first approximation to sunshine